today. I'm going to be telling you about work that my lab has been doing to develop gene co-expression network analysis and these network analysis tools to identify gene candidates for specialized metabolic pathways. And so this sort of analysis using co-expression to identify gene candidates is more often referred to as guilt by association, which I think it comes from its first being used to study disease-associated genes. So you see this researcher here with her pipette in hand, trying to pick out some gene candidates from a lineup. And here is what it looks like in practice, where you have some gene, like gene 3 here, co-expressing, so behaving very similarly to these other 4 genes known to be associated in some disease phenotype. And so identifying gene 3 as a potential gene of interest. However, for our purposes, I think that honor by association is much more appropriate when we think about specialized metabolism and how important these pathways and products are for not just the ecology, but the evolution of plants, providing resistance for different biotic interactions, and also their numerous applications for humans and agriculture and medicine. I think honor by association is a much more appropriate term. But in general, whatever you call it, one of the things that we think about in the lab is how generally can we apply honor by association to metabolic pathways. And so here I'm going to show you 2 contrasting examples. In the first, we'll look at 5 genes involved in glycolysis, just 5 genes here, and we'll look at their gene expression over 300 different RNA-seq samples taken from a public database. And here you see that the genes expression are not very strongly correlated with each other. So some gene pairs may be positively correlated, so this GAPC1 and PGK, but some are also pretty strongly anti-correlated, FBA1 and PGM. But then others show no real strong pattern, either positive or negative in terms of their correlation. And so if you contrast that with 5 genes involved in glucosinolate biosynthesis, which is a specialized metabolic pathway really important for plant defense in Arabidopsis and other Brassicaceae and Brassicales, you see a much different pattern. You see very strong pattern of correlated expression, also shown here in this bright red heat map. Another way to look at this would be to show the entire, zoom out to the entire pathways and look at them in terms of a co-expression network graph. And so here the circles represent genes, and the stronger the co-expression between 2 gene pairs, the closer those 2 genes are going to be showing up in this network graph. And these 2 pathways, glycolysis and glucosinolate biosynthesis, show 2 examples in this larger pattern that specialized metabolism, like glucosinolates here, tend to be much more transcriptionally co-regulated compared to core pathways, as demonstrated by them forming a very tight network with an average correlation of 0.44. And so my lab is interested in investigating how we can tailor gene co-expression analyses to target specialized metabolism pathways specifically, and not worry so much about the larger global network, which includes the large hairball of the global co-expression network that consists of mostly core metabolism and other things. So to do that, go after patterns in the network that look like glucosinolates here. So identify small modules of genes, fewer genes, shorter edges between them that are behaving very similarly to each other and different from the rest of the global network. And so to do this, my lab has developed this, what we're calling the mutual ranks to modules workflow. That I don't have time to get into the specifics of the methods, although I have the slides saved for later. If people want to get into the methods and more of the question and answer section, I'm happy to. But very briefly, we perform a series of transformations. So start with our Spearman's correlation of co-expression, transform those to mutual ranks, and then again to get an edge weight transformation and use those modified edge weights to call co-express gene modules. And we have all of this outlined in a series of tutorials that are available on GitHub if you wanted to check that out. And so what we found, if we look at Arabidopsis, for example, is that the mutual ranks to modules workflow does a very good job of recovering experimentally validated pathways like glucosinolate biosynthesis here. So what you see are, this is a 43 gene module, and 23 of them, shown in blue, are genes that are known to participate in glucosinolate biosynthesis, and it represents the majority of genes in the pathway. Those are the blue genes that co-express with each other in our modules, shown in this pathway schematic. Although you will notice that there's one outlier here, GGP1, shown in black instead of blue, because it was never, we could never get it to be recovered in any of our co-expression networks. Although we do have DJ1F, which is a homolog to GGP1. They are both glutamine aminotransferase-like proteins. And so you see it is very strongly co-expressed with other glucosinolate genes. And I was a bit surprised to dig into this pathway in more detail. It's not my background initially, but I learned that GGP1 has never been demonstrated to participate in glucosinolate biosynthesis in planta. It was flagged as a candidate based on microarray data and its heterologous activity in tobacco. And so DJ1F has been perhaps overlooked because it was never included in that original microarray chip. And so this is something that a postdoc in my lab, Dr. Josh Trujillo, is interested in investigating to see if DJ1F, rather than, or perhaps in addition to GGP1, is participating in glucosinolate biosynthesis in Arabidopsis. So this is an overview of the sort of results you can get in a model organism, model system of glucosinolate biosynthesis, where we have a lot of known glucosinolate genes. And we also have a giant RNA-seq data set, over 1,400 samples going into our global co-expression network analysis. For those of you working on non-model systems, or a pathway that is a lot less studied than glucosinolates, you might be wondering, now, how many samples are we talking about here? What do I need to put together in order to apply the same sort of analysis to my system of interest? And so I have at least an anecdotal story that I think may interest you, that comes out of a collaboration between my lab. Most of the work I'm going to be showing here is done by Bob Auber, a graduate student in my lab, in collaboration with Josh Woodholm at Purdue in horticulture and landscape architecture, and his grad student, T.T. Sitiot. So what we've been investigating, right when I started back at Purdue in 2017, we started this project looking at this Shikonin biosynthesis. It's a pathway. Shikonin is a specialized metabolite shown. It's a red pigment here, an allelopathic chemical produced by boraginales, including the Lithospermum erythrorhizin. And this red pigment, so you can see it in like this cross section of the root here. This red pigment has been used for pharmaceuticals, or I should say, dietary supplements and cosmetics. And recently it's been highlighted, its function, its activity as an anti-inflammatory and other drug activities has been of interest because it's been shown to perhaps suppress HIV and breast cancer activity. So there's lots of interest on the applied side of trying to get to the bottom of the Shikonin biosynthesis pathway, trying to metabolically engineer it to perhaps increase Shikonin production or apply synthetic biology approaches. but we're hampered by the fact that we don't know much about how this pathway, the genes that actually underlie this pathway in the plant itself. So up until recently, only this prenyltransferase PGT, shown here, was the only identified enzyme and gene that had been linked to Shikonin biosynthesis. More recently, kind of as when the last few years, when we were already kind of teasing apart this project, an additional cytochrome P450 was identified that can act on this geranyl hydroquinone substrate later on in the pathway. So very little, there's most of these steps are hypothetical with unknown genes associated with them. And so the first thing we did in the lab was sequence the Lithospermum erythrorhizin genome using both Illumina and nanopore long reads. And that paper came out a few years ago. And with that genome in hand, we performed a comparative phylogenomic analysis looking at these 4 species, including our new genome, that was able to produce Shikonin. The other Baraginales shown here do not produce this compound. And so looking at what sort of genes would have duplicated in this last common ancestor of Shikonin producing species, we identified almost 4,000 gene families. But looking within that family, those families, to see what sort of genes was in that set, and if any of them look like they could be participating in Shikonin biosynthesis. Just one of those genes that we found interesting was the gene family that contained this PGT enzyme. So canonically, or I should say ancestrally, there was a single copy of this prenyltransferase gene family involved in ubiquinone biosynthesis. But you see there's been multiple rounds of gene duplication in the Baraginales and Baraginaceae, including this retrotransposition duplication event, and then subsequent radiation of that gene, leading to PGT1 and 2 are 2 enzymes that are known to participate in Shikonin biosynthesis. PGT1, I'll say, is shown to be the dominant player in the pathway in planta, at least in our experiments. But so you see that gene duplication playing a role in the evolution of this specialized pathway. Now that we have our genome, what could we apply that same sort of co-expression network analysis that I showed you in Arabidopsis to this species and this pathway? And here we had a far fewer number of experiments to work with. We only had 3 discrete experiments, 14 total RNA-seq libraries that the Whitholm lab had already generated as part of their work. And in these experiments, you can see that although there's not very many of them, they are very they're not selected at random. They were selected in order to identify genes that are participating in this pathway. So we have samples from whole roots where Shikonin is abundant, and this PGT gene shows high relative expression, and then samples from leaves where the Shikonin is not produced and PGT is not expressed. Similarly, if we zoom in on the root, we can take samples of shaved periderm, the outside of the root where the Shikonin is present, compare that to vascular tissue where it's absent. And inside hairy root tissue cultures grown in the dark where we have Shikonin versus in the light where Shikonin is not produced. And you can perform a traditional differential gene expression analysis with these data and look at the intersection of all these experiments. And from that, you see 374 genes that are up in all conditions where Shikonin is produced, including the PGT1 gene that I went over earlier. But you'll see that 374 is still quite a lot of gene candidates to be working with. And not only that, our cytochrome P450 that had been identified kind of as we were working on this project wasn't recovered in that set. It was not differentially expressed in the root versus aerial tissue. So It was different, just not significant. And so that is kind of a good example of how differential gene expression analysis for this sort of experiment can kind of yield a lot of not just false positives, but some also false negatives too, and leave you a lot of candidates to kind of sift through. Versus if we take those 14 RNA-seq samples and run them through the mutual ranks to modules workflow, you wind up with a module like this, where we pulled out all modules where that contained the PGT gene of interest. And here you see that one shown with this asterisk, the red gene with the asterisk, and these other red genes are other PGT like genes in that parental transferase gene family. We also see the cytochrome P450 here that we already knew to participate in the pathway, as well as some additional P450s of interest, some transcription factors and transporters that may be of interest for future investigation. And in pink, there are genes for the MVA and the phenylpropanoid pathway, which provide precursors for Shikonin biosynthesis. So all of that makes sense. The one gene that the Whithelm lab was particularly excited about was this FPPS like gene here shown in yellow. And the reason why that interests us so much is because there's two alternate routes to isoprenoid biosynthesis. There's the MVA pathway in the cytosol that takes the IPP precursor, and through farnesyl pyrophosphate synthase produces this 15-carbon FPP intermediate. And then there's the MEP pathway in the chloroplast that produces, with the geranyl pyrophosphate synthase, produces this 10-carbon GPP intermediate. But Shikonin's kind of weird. It is produced in the cytosol via a GPP intermediate, but labeling in the 70s showed that that GPP precursor was derived from the precursors from the MVA pathway. So what exactly is going on here? There's two alternatives. It could be that a gene from the GPPS family has evolved novel targeting and is now functioning in the cytosol. Alternatively, you could think that the FPPS gene has evolved novel GPP activity. And so that is what seems to be happening here when we see this GPPS-like, that will happen, FPPS-like gene showing up strongly co-expressed with our PGT gene that we know is involved in Shikonin biosynthesis. So we performed a phylogenetic analysis of the FPPS gene family in green plants, and I'll focus on the canonical FPP synthase clade first. So here we have 3 genes in Lithospermum that fit in this canonical clade. None of these genes co-express with PGT, and they are sister to known taxonomic neighbors, how we would expect this gene family to behave, grouping next to the solanales and the laminales. And these genes likely maintain the canonical FPPS function to work in producing other known isoprenoids. In contrast, the second FPP synthase clade contains that gene of interest that I showed you in yellow that co-expresses with PGT. From now on, I'm going to refer to it to the GPPS in Lithospermum. It's a little confusing, but this gene co-expresses with PGT, and you'll notice that the genes in this clade all contain a His residue before the farm motif, or the first aspartic acid rich motif that's thought to influence product specificity. So we thought that this was very interesting, and then another group beat us to the punch, unfortunately, and was able to perform site-directed mutagenesis of this region and show that the His residue actually does determine product specificity leading to GPP in lithospermum for Shikonin biosynthesis, which was great because they've saved us having to do that experiment ourselves. TT in Josh's lab followed up with an RNAi experiment to show that when we knock down this GPPS gene in lithospermum, we also knock down Shikonin production. So all of that together shows that we have a novel FPPS homolog that has evolved GPPS activity and is responsible for the production of this GPP precursor in Shikonin SM pathway. And so I've added it to our list of known genes here for this pathway. We haven't stopped there. This is still kind of a work in progress. We have some additional genes that are of particular interest to us, and I'll just kind of go over some of them. We have that were also showing up as co-expressed in our network analysis. We have a prephenate dehydrogenase that catalyzes an oxidative decarboxylation reaction, canonically in tyrosine metabolism, but we're interested if this paralog, this duplicated PDH gene could have evolved to act on another 4-hydroxylated substrate and be participating in this decarboxylase reaction here. If you recall from that phylogeny at the beginning, I mentioned that PGT also evolved via that retroduplication from a gene involved in ubiquinone biosynthesis. And interestingly, we also identified several other ubiquinone gene paralogs co-expressed with these 3 genes in Shikonin biosynthesis. So we're going to be exploring that in a little bit more detail. And lastly, we have some acetyltransferases that are interesting to explore as potential tailoring enzymes, perhaps involved in synthesizing some Shikonin derivatives. So with that, I hope that what I was able to show here today is that we can apply honor by association to metabolic pathways, even those in which we have a very minimal RNA-seq data set to work with. Provided that we have designed that RNA-seq data set using prior knowledge of the pathway. So if we can select conditions where our pathway of interest is where the regulation of that pathway is different between the different conditions, that we can increase the power of our analysis, even if we have a limited data set. So with that, I would like to thank all the members of my lab and my collaborators, in particular Bob here, who performed a majority of the work that I showed you today. And then here is my GitHub page for the MR2Mods pipeline, if anybody wants to check it out and see about applying it to their own data sets. So with that, I am happy to take any questions.
Thank you very much, Jen. It's a great talk. Yeah, for questions, please feel free to raise your hand or post your questions into the chat and we can read them. Benjamin, yeah, please go ahead.
Hi there. Yeah, really nice talk. This duplicated FPPS does this clade, is it just kind of with the family of plants that you're working on, or is it quite a deep duplication?
Yeah, so let me share that again, or maybe we can just zoom in, leave it on. So that's a really good question. So we focused on the duplication in the Baraginales. So our duplication seems to have occurred in the ancestor of the Baraginales. Not all of them make Shikonin. So only the red species here are producing Shikonin. This duplication seems to have preceded the evolution of this metabolite. but it's cool that you mentioned that, because we do see additional duplications of the FPPS gene family. So I've actually starred them here. There's some duplications. There's a duplication in strawberry. This is a Asian pear, and also in tomato. I'm sorry, potato. These genes are all there are paralogs of the FPPS genes in these species, if that makes sense. There are multiple copies of this gene family in all three of these starred species. And the reasons why I starred them is one of those copies has evolved this His residue, which makes us suspect that perhaps the same sort of thing has happened multiple times in other species, but we haven't looked into it. And our gene family, our phylogeny definitely focused on the Baraginales. To dig into these specific events, we would want to redo our phylogeny and add additional closely related taxa for these guys.
Great, thank you very much.
Sure.
Cobus, please go ahead.
Hi, Jen. Nice talk, really interesting. I love this type of methodology approach with networks. I was just wondering if you can comment on how this approach is different to like a WGCNA approach. Where you, yeah, if you can comment on that.
Yeah, I thought about throwing in like a slide to compare it. And then I figured we could maybe save it for talk. So this is perfect. There's a few major differences between our pipeline and WGCNA. So WGCNA kind of does this it constructs a dendrogram of all of your genes and kind of draws lines where it's gonna all genes like downstream of when it bins the different colors. I'm doing a horrible job of explaining it, but basically you have genes assigned to one module or another in WGCNA, and all genes are assigned to a module. And so it very much just tries to put circles around the entire global network and bin every gene into a module. And so what we did when we were kind of designing our workflow is instead we said that because we're really only interested in specialized metabolism and we want those really tiny, smaller, discrete modules, we used a different type of module calling program. It's called Cluster 1. And so here is an example of what it can do. It can bin genes into multiple modules. So here in this, can you see my cursor?
Yeah.
Yes. Okay, great. So here is a gene that has been binned into module 3 and module 4 because it's kind of in the intersection of those two overlapping modules. And then a lot of genes, I should say, are not assigned to modules at all. And so in that way, we're kind of setting aside a lot of the genes involved in like core metabolism and that form like the densest part of that network hairball. We're just setting them aside and we're not even looking at them. And we're only looking at these kind of discrete modules that kind of form the periphery of the global network. The other thing that is nice about, or that I think our program does differently is works with mutual ranks. What do I have here? There's been some work done with mutual ranks showing that when you transform a Spearman's or a Pearman's correlation to mutual rank based, that you are doing that metric is better able to recover like pathway level modules. So just another thing that I like about the mutual rank metric versus, I believe WGCNA mostly uses the Pearson's, correct? Other people are more familiar with it, but yeah, those are a few things that we do differently.
Thank you.
Hi, Jen. So
Hi.
the same question you already answered most of what I wanted to ask. In addition to WGCNA, I was wondering if, since this is a fuzzy approach, have you compared this with fuzzy k-means or some other, like, is there a citation which, or a study which says that this approach would be better at finding true associations versus
Yeah.
positive, negative?
I can't remember if the paper, so I'd want to go back and look at this 2018 paper. If they looked at the fuzzy k-means clustering, I'm not sure. I can't remember, but this is definitely, I will also say for people who are kind of interested in methods that I'm not a computational biologist. I don't work in developing algorithms. And so part of, I've co-opted this cluster one approach because I liked these, it very much fit the sort of modules that I was trying to call, and it performs pretty well in practice. But I do think that this is a place where improvements could be made, like, if that makes sense. I feel like there's a lot of room for maybe incorporating like machine learning algorithms, taking in other sources of information about a gene's characteristics for specifically going after specialized metabolic modules from global co-expression networks. It's the calling the module spot, drawing the circle around your set of genes, that I do think that there's a lot of room for kind of improvement and program development there.
I agree. So in my own experience, WCNA gives some of the modules are really large,
Yeah.
hundreds of genes, and that's biologically meaningless. But mutual rank does give smaller, tighter modules, so that helps in interpretation.
Yeah. Yes.
Okay, we have another question from the chat. What do you consider more important for predicting co-expressed genes? Biological information, an example, more tissues and or conditions, or technical RNA-seq replicates with less samples?
I say if you're going to put me on the spot and for me to say, I would say more conditions, more tissues, more conditions, experiments, etc. The technical replicates are in some ways a lot less necessary for this co-expression analysis. So the replicates are for doing the differential expression so that we can say with confidence that, like, this gene is behaving differently between these two conditions. And in the co-expression, we kind of care less about what the gene is doing in any individual sample and more about what it's doing across the total experiment. And so in that way, the more variation in gene expression you can introduce to your network, I think the more robust the analysis will be as far as pulling out discrete, like, signals for different pathways. Although I will kind of go off on a little bit of a tangent and scroll back up to our project design. Here we did have 3 replicates, technical replicates for periderm, 3 for vasculature. And I thought this was very interesting because here, this is very much technical replicates were variable, because this is a person shaving off in the Whithelm lab, shaving off the outer periderm and just deciding kind of when to stop. And so there was definitely some variation in how much did some additional vasculature tissue accidentally get carried over into some of the periderm, etc. You could know that there would be some noise in the technical replicates of this experiment, but that noise then is still signal in a co-expression analysis, right? So that if that makes sense. So it kind of depends on your pathway that you're interested in, but my default reaction would be to try to get as many conditions as possible, and less so on the replicates. Although replicates are helpful for knowing whether or not you have done something horribly wrong, I suppose, in your experimental setup or something.
Great, thank you. Do we have any more questions? Otherwise, I have a question directly to the slide. I was just like found it interesting that you saw this dark and light induction of the Shikonin pathway. I was wondering what kind of regulatory mechanism is behind that, or is there any hypothesis?
That's a good question. I bet Josh Woodhelm could answer your question better, because I don't know. I can say one of the things we did look at in our experiments that is in the paper, but I didn't discuss in the slides, is that Bob performed a motif analysis of the upstream promoter regions of the module genes and found an enrichment in a motif for worky transcription factors that we did find. We do have some of these blue transcription factors are also worky transcription factors. So there might be one. So that's a dodge because I don't have any like specific biological information on how this pathway is regulated in the light and dark. But that are sort of analysis. One thing you can do with this sort of analysis is look for upstream enriched regulatory motifs and try to get at pathway regulation through these sorts of pathways, this sort of analysis as well.
Yeah, thank you. That's an interesting thing.
Yeah, sort of an aside from your actual question, but yeah.
Thank you. Okay, if we don't have any more questions from the audience, then I would like to thank everybody for